Hello students, today we'll learn about determination of cell viability by dye exclusion assay using trepan blue. Introduction. Cell health is indicated by the number of viable cells in the culture or the rate of proliferation. As cells under culture can be exposed to various chemicals and physical agents that can alter cell health and metabolism, assessing viability of cells is necessary for routine maintenance as well as for other experimental purposes. Physical and chemical agents can cause toxicity to the cells through different means such as membrane destruction, blockage of metabolic pathways, blockage of protein synthesis, prevention of DNA synthesis, or binding to cell death receptors. There are different methods developed to ascertain cell death and viability. Trepan blue staining is one of the cell viability assays that is cheap, reliable, and easily reproducible. Cells that are cultured in vitro are used for various experiments such as in drug screening or evaluation of cytotoxicity of certain chemicals. Since they do not require the use of animals and are relatively inexpensive, as well as it enables screening of large number of drugs, the technique is being employed in wide area of research over recent years. In cancer research, the technique is being used to evaluate efficacy of drug during drug screening and development. Cytotoxicity and cell viability assays are based on various cellular functions such as compromised membrane integrity, enzyme activity, energy production, and nucleotide uptake function, just to name a few. There are certain advantages of in vitro cell culture and viability assays such as reduced costs, rapid determination, as well as potential for automation. Moreover, human cell lines can be used for drug screening, which is more relevant than using animals. However, the method is not so advanced enough to replace the use of animals. Since it is important to determine the number of viable cells at the end of a cytotoxicity test experiment, cell viability assays presents a quantitative data that can be used for statistical analysis to substantiate experimental results. There are numerous cell viability assays to choose from depending on the principle and interaction type they are based on. Now let's discuss about classification of cell viability assays. Cell viability assays can be categorized as: number 1, dye exclusion assays such as trepan blue, eosin, Congo red, and use of erythrocin B. The second type is the colorimetric assays, which are XTT, MTT, WST, MTS, SRB, crystal violet, NRU assays. The third type, which is the fluorometric assays, uses LMR blue and CFTA-A assays. The fourth type, which is known as the luminometric assays, uses ATP assays. Now let's discuss more about the dye exclusion assays. Dye exclusion assay is one of the simplest and rapid method for determination of cell health. Viable cells have an intact membrane and tend to exclude dye from entering the cell cytoplasm, whereas apoptotic or necrotic cells have compromised cell membrane that can no longer prevent entry of the dye into the cell cytoplasm, and hence cells stained with appropriate dye are cells that are dead or near death. However, the method of dye exclusion has its own disadvantages, as processing of a large number of samples is not possible. Some of the dyes that are used in this assay are Congo red, eosin, erythrocin B, and trepan blue. Among them, trepan blue is the most widely used. While using dye exclusion assays, certain factors may be accounted for, such as damaged cells requires few days to lose membrane integrity. Secondly, cells that are still surviving will continue to proliferate. Some lethally damaged cells may not be stained by dye exclusion due to early disintegration. Here, the second factor and the third factor may be a cause of underestimation of cell death when data is to be prepared as percentage of surviving cells. Now, we will discuss about the principles of trypan blue dye exclusion assays. Trypan blue is a negatively charged dye and has affinity for the positively charged parts of a cell. As mentioned earlier, this method is based on the fact that live cells possesses intact cell membrane that exclude dye, whereas dead cells have permeable membranes that cannot exclude the dye from its cytoplasm. After exposure of the cells to various doses of test compounds and incubation for a certain period of time, the cells are harvested after proper washing off of test compounds. Cells are suspended and a portion of the cell suspension is mixed with dye followed by examination under a light microscope. Viable cells will have a clear cytoplasm, whereas dead cells will have a blue cytoplasm. After counting the number of viable cells and dead cells, percentage of viability can be determined. There are certain advantages of using trypan blue. Trypan blue dye exclusion assay is a relatively simple, inexpensive, and rapid method for determining cell viability in cultured cells. However, the disadvantages are cell counting requires a hemocytometer and hence counting errors can occur. Poor handling resulting to poor dispersion of cells can result in miscalculation of number of cells. The use of hemocytometer also warrants handling errors such as improper filling of chamber, presence of air bubbles, as well as inaccurate dilution of cells. The procedure also has limitation of processing large number of cells. Moreover, the method cannot distinguish cells that are healthy and those losing viability, and hence this method is not sensitive enough for in vitro cytotoxicity testing. In materials required, we will be requiring cultured cells, phosphate buffered saline, centrifuge tubes of 15 ml and 1.5 ml, 0.4% trypan blue, light microscope, hemocytometer, pipettes, pipette tips, and 0.25% trypsin solution. In the procedure, to preparation of cells for determination of viability. In the first step, remove the culture medium from the culture vessel by aspiration. Then, wash the monolayer with PBS, which is free of calcium ions and magnesium ions, to remove the serum. Next, aliquot enough trypsin EDTA solution into the culture vessel, completely covering the monolayer of cells, and place in 37 degrees centigrade incubator for approximately 2 minutes. In the next step, the cells with trypsin are allowed to incubate until cells detach from the surface. Progress of trypsinization can be checked by examination under an inverted microscope. Here we should note that as trypsin can damage cells, overexposure should be avoided. Cells of different lineage may require different incubation time. In the fifth step, when trypsinization process is complete, the cells will be in suspension and appear rounded. In the next step, add serum or medium containing serum to the cell suspension as soon as possible to inhibit further tryptic activity which may damage the cells. Now, centrifuge the trypsinized cells at 2000 rpm for 5 minutes to pellet down the cells. Next, resuspend the cells in 10 ml of complete growth media. For determination of cell viability, in the first step, prepare a 0.4% solution of trypan blue in buffered isotonic salt solution, which is of pH around 7.2 to 7.3. This may be a phosphate buffered saline. Next, add 0.1 ml of trypan blue stock solution to 0.1 ml of cells. Load a hemocytometer and examine immediately under a microscope at low magnification. Count the number of blue staining cells and the number of total cells. Cell viability should be at least 95% of healthy log phase cultures. In the results, cells stained with blue and unstained cells will be observed. Those stained with trypan blue are non-viable or dead cells, whereas those that are unstained are live cells. In order to calculate percentage of viable cells, the following formula is used. Percentage of viable cells will be equal to 1 minus number of blue cells divided by number of total cells multiplied by 100. To calculate the number of viable cells per ml, the following formula can be used. We should always remember to correct for the dilution factor here. Number of viable cells multiplied by 2 multiplied by 10 to the power 4, which will be equal to cells per ml of culture. Here, number of viable cells are multiplied by 2 because the cell suspension was diluted twice when we took equal volume of cell suspension and trypan blue. In conclusion, we have understood that cells that are intact and live are not stained with trypan blue as the dye is unable to enter the cell cytoplasm, whereas dead cells and cells that have compromised cell membrane will stain blue. Among the various means of determining cell viability, trypan blue is the simplest and inexpensive method but has limitations as it is not suitable for large-scale analysis. Thank you.